What's up, Psychedelic Spotlight? I'm James from the Psychedelic Investor, and for today's podcast, we have a special one for you. And that is because we have the one, the only CEO of Bright Minds, Mr. Ian McDonald, on the line today. Ian, welcome to the channel. How are you doing today?
What an intro.
Hi.
I'm doing great. Thanks for pumping my tires there.
Oh, no worries. I've just been sitting in my chair practicing it for the last 10 minutes, so I hope it came out properly.
Oh, no, it sounded great.
Perfect. So I guess my first question for you is, tell us a little bit about the vision of Bright Minds, and what are you guys trying to accomplish at your company?
So Bright Minds really started with the idea that first generation psychedelic medicines, while effective, are not in their final form. We felt that we could improve upon natural compounds like psilocybin and other first generation compounds like LSD, DMT, etc., in a number of areas that would make them more, that would accentuate more drug-like characteristics. Now, what do we mean by this? Specificity in terms of target receptors. Some of the other rough edges that first generation psychedelics have would be oral bioavailability in the case of DMT or 5-MeO. And then taking a look at some of the other psychedelics, LSD, psilocybin, it would include in these would be time. There's some other more, I would say, complex areas that we're working out as well that we probably don't have time to get into, such as biased agonism. But those would be some of the main areas of improvement that we're looking at targeting, and really building upon this first generation of psychedelic medicines. In order to do this, we needed a team that specialized in not only the biological understanding of psychedelics and how they work in the body, but also the medicinal chemistry aspect. So biology is, in layman's terms, how the drugs interact in the body, and medicinal chemistry is actually going there and designing new molecules, putting an atom on here, taking one off there, to make sure that they fit as well into the specific receptor you're targeting without targeting other off-target receptors.
Okay, so that's very interesting. I think I'm just going to repeat what you said to make sure that I have a solid understanding. So over the past couple of years, there's been lots of great evidence that have been coming out about psychedelic compounds like LSD, like psilocybin, like DMT, in regards to how they can be helpful with mental health care treatments. But despite some of these benefits, there's also a lot of potential negativities when it comes to how they interact with their bodies, maybe side effects. So what you guys at Bright Minds are doing are, instead of working with the classic psychedelics, the psilocybin, the LSD, the DMT, you're working on kind of second and third generation psychedelics, tweaking the chemical formulations of these compounds so that when people consume what are essentially now brand new drugs, new chemical entities, they are more tailored to the effects that you want to see out of the therapy setting. Is that kind of it?
Yeah, that's totally it. So this is typically the progression you see in pharmaceutical drug development, whether it's in psychedelics or whether it's in other areas of medicine. You saw this with traditional antidepressants. The first generation was something called tricyclic antidepressants, which worked on a number of different targets in the brain. They had quite a lot of side effects. Those were then usurped by a second generation, and eventually you had SSRIs, which were more targeted therapies that eliminated a lot of those negative aspects. And you see the same thing in you've seen the same thing in antihistamines, which would be allergy medication. The first generation there, they worked, but they made you very sleepy. So what medicinal chemists did was design drugs that didn't cross the blood-brain barrier as much as those first-generation drugs did. Now today, those first-generation antihistamines are actually the over-counter sleeping pills. And if you have allergies and seasonal allergies, you typically wouldn't take those drugs if you were trying to go about your day and not fall asleep.
So how is it that you guys come up with these new drugs? Are you using computer algorithms to try to work through like a million different combinations? Are you just throwing a bunch of chemicals together in a lab and injecting it into a rat and see what happens and then kind of move on from there? Or what's the kind of process for creating essentially a medicine that's never existed before?
Yeah, so I think it's a mix of a couple of these strategies you mentioned there. So we're working on a couple of different chemical scaffolds, and a chemical scaffold is just a fancy way of saying what does the basic structure look like? Does it look like psilocin? Does it look like LSD? And we're working on structures that look somewhat like those chemical backbones. But we're also looking at novel structures. So these are completely different psychedelic classes that have that are known and that we're really pioneering. So
would an example be like, I'm just trying to understand this right now. Let's say you want to have a drug that affects a specific receptor in the brain. So the one that a lot of people on this channel know about is the 5-HT2A receptor. So would you have kind of like a scaffolding of your drug that you want to affect that, and then you have all sorts of different variations of it? So you have like different blanking on my science words here, but different isomers and stuff of the same compound, and you would have a whole bunch of different ones that you can then try in computer models or maybe rats or something?
Yeah, exactly. So our first step is taking a look at the literature, see what worked, see what didn't. We have molecular models of the serotonin 2A receptor, and we dock various chemical entities into that structure, and we see, okay, is it hitting this part of the receptor, in which case there may be a covalent bond, for example, or is it hitting this part of the structure? Is this part rotating? And we get an idea of how well that drug not only fits in the receptor, but its potency as well. So that allows us to get a pretty good understanding, and going back and taking a look through the psychedelic literature. And by the way, I think we have probably one of the largest libraries of psychedelic drugs, because a lot of the literature was actually inaccurate. We went back and then basically did it over from scratch again. So you learn what works, what doesn't. Okay, does this atom here add to efficacy? Does it add to potency, or does it create an inactive compound? And we take those learnings, and it's really an iterative process with our medicinal chemists and molecular modelers. So that's the first area of triage. And then once you have compounds that you're generally quite confident about, then you move into chemical synthesis. And from chemical synthesis, we do actual receptor binding. So we'll take cell lines, and we'll take our compound, and we'll see how well those new compounds activate those specific cell lines that we're targeting. So a cell line would, for example, be a serotonin 2A receptor, or a serotonin 2B receptor, or a serotonin 1A receptor. And from that point, once we've seen what we're looking for in terms of receptor binding and that baseline of pharmacology, then we go into screening studies to check for any potential liabilities, like a hERG liability or a cancer signal, etc. If it passes that, then we move into rodents and behavioral assays. And
roughly, how big would you say your library of different compounds is? Are we talking you have 10 different compounds, more?
Oh, we've made hundreds.
Hundreds of different compounds. Okay.
Yeah.
And
we have as we speak on our psychedelic program, we have, I'd say, around 25 chemists working for us.
Okay.
And these are just chemists. These are guys that's gotten to the stage where, like, past molecular modeling, we're relatively confident in these compounds, and we're creating the physical matter to test in these receptor binding models.
Very interesting. So let's talk about a specific compound. The compound I want to talk about, and then also your views on it, is psilocybin. So recently, there have been quite a few major studies come out that show that psilocybin is very effective in treating depression, among other things. But when I'm thinking about depression, there was one study that showed it was 4 times more effective than an SSRI. There was another study that I believe was done by the Imperial College of London, which showed that the remission rates for those that were on psilocybin were twice as high as those that were taking an SSRI. But at Bright Minds, you guys have a few issues with the compound. So I was wondering if you could just walk me through what some of those issues are and what you guys are doing differently.
Sure. So psilocybin is a great starting point. Mother Nature's done a very good job, but unfortunately, in our view, it's not a perfect compound. It could be made better. There's no question that
I feel like that's the tagline right there, what you just said. Psilocybin, oh shoot, how did you phrase it? Psilocybin is a great compound. Mother Nature did a great job, but it's just a starting point. Like I love the starting point kind of mentality. Sorry for cutting you off. You can continue.
Yeah, and I mean, if I can just use if I can just quickly go into an analog of this. If you take a look at one of the most commonly used drugs in the world today, aspirin, that started off as an extract from willow bark. So people would take willow bark off a tree, they would brew it. It would be a tea. You would drink that tea, and it would have anti-pain properties. And over time, it was elucidated that there was a particular compound in there that was responsible for that. The issue was that compound was not perfect. It was very difficult to digest, and there were all sorts of stomach problems that came along with that. And then a chemist around the turn of the century started working on this, doing pretty much the exact same thing that we're doing now. We have the advent of modern day technology, but other than that, the same basic concept, which is designing a compound that could have the positive effects without the negative effects. So he created aspirin, and today I don't think you see anybody brewing willow bark to treat a sore knee or a backache. So
I'm sure there's a couple people out there in the wilderness still doing that.
There may be, and I think there's still going to be people taking mushrooms 30, 40 years from now. We're all for that. What we're trying to do is create a drug that gives prescribers a safe, reliable experience that is really tuned to treating depression and related illnesses on a large scale. So the trip time of psilocybin, I think, is not optimal. We believe that 4 to 6 hours of call it intoxication of
tripping ball
of trip is more than is needed for you to get that brain reset effect. So what we're doing is creating a drug that gets absorbed quickly and metabolized quickly. You need to meet a threshold to get that reset effect, but after that, you don't really need the drug to stick around. So in order to treat millions and millions of people, we are tuning that PK down, creating a shorter half-life compound. We're also working on some of the other issues, such as 5-HT2B activation, which in large patient populations has been shown to cause cardiac valvulopathy. Let's
just back up there a second, just so that everybody understands that. So
sure.
One of the main effects that psilocybin has that has been shown in these studies is how it's relating to the 5-HT2A receptor. But a lot of people don't really know or understand that it affects other parts of the brain too. And what's in some of the literature that you guys at Bright Minds have put out is the idea that the fact that it affects the 2B receptor might not necessarily be wanted or desired outcome for some sort of medical benefit. So could you just maybe explain a little bit what the differences are between 2A, 2B, and maybe 2C? Because I know that you guys are also, and that's a good segue for the next section of this interview, talking about focusing on the 2C enzyme in the brain.
Yeah, sure. So 5-HT2A is the molecular target for all psychedelic drugs. It is activated by all the ones you know and love. Ketamine would be one that isn't, that works on the NMDA receptor, but all serotonergic drugs, serotonergic psychedelics, to some degree activate 5-HT2A. That is also the target of the antidepressant effects. Now, psilocybin is quite unselective in its approach. It's a dirty drug, as we generally say in biotech. It hits a lot of receptors, meaning that it hits that 5-HT2A receptor, which you want it to hit for the psychedelic and therapeutic effect, but it also hits the 5-HT2B receptor, which is a major cause of concern. So the 5-HT2B receptor has been linked to cardiac valvulopathy, which is just a fancy way of saying hardening of the heart valves. And that the only way to cure that, unfortunately, is a heart transplant. Now, this isn't going to happen with everybody. It's going to happen with a very small subset of the population over a long period of time. But if you can tune that out, it's sort of a no-brainer in terms of making a safer drug. So
it's kind of like, to give a metaphor to it, using psilocybin for effects in the brain is kind of like a shotgun that goes everywhere, whereas you want a sharpshooter to target one specific receptor. Okay.
Yeah, exactly. It's very unspecific in its action, which just floods the brain with serotonin, which is good. But if you can have a more targeted therapy that hits the receptor that you want and avoids the other ones that aren't involved with that therapeutic action that you're creating the drug for, then you have something that can be used for specific patient populations. So 5-HT2A, depression, PTSD, all that good stuff. 5-HT2B, bad guy, we want to avoid that. 5-HT2C is another receptor target, which we're focusing on at Bright Minds, and creating agonists or compounds that activate this receptor have application in a few different areas. So this is a non-psychedelic. This would be a daily drug, like your standard pharmaceutical. And 5-HT2C works to help with illnesses such as really all forms of addiction, or what we've seen to be all forms of addiction, everything we've tried. Nicotine, opioid self-administration, cocaine, we believe it would work for. It would probably even work for things like social media addiction and
the behavioral addictions also.
Yeah.
Okay.
Yeah, it really works with impulse control disorders. So we tried it in binge eating, which is more or less food addiction, and it could potentially be used for something like Tourette's or OCD, and the common bond there is really an impulse control.
So would you have one drug to try to affect or conquer all of these different impulse controls, or you would fine tune it for, say, like binge eating as compared to a cocaine addiction? They would be two different drugs, or it'd be the same one just used for all sorts of different impulse control issues?
Yeah, the same one would work for all of those.
Interesting. I'm going to shift gears here for a second, because one of the most interesting areas of your drug development platform, or the areas in which you're trying to ameliorate, is essentially pain treatment. Because when a lot of people think about psychedelics companies or psychedelic inspired companies, they don't necessarily think of pain as something that you would want to, or that it would be beneficial to treat. So I'm just wondering if you can give me a little bit of the philosophy behind why you want to treat pain, and then also how you're planning on treating. Is this a 2C thing, or is this a 2B thing? Sorry.
We believe a mixed agonist profile would be best suited for these indications, meaning that it hits both the 5-HT2A and the 5-HT2C targets. Just to give your audience a little background on the current standard of care for pain, you have NSAIDs, which are the chemical class of drugs like aspirin, all the real over-the-counter ibuprofen, et cetera, all the over-the-counter pain medications that you would know. Those drugs are, that's one group, and the other group are opioids. There's really nothing else to treat pain. So if you have a chronic pain indication, it could be fibromyalgia, it could be a horrible accident you got, a horrible car crash you got in, and your back's never been the same, or a workplace accident. You don't really have many options. NSAIDs are good for call it mild to moderate over a short period of time, but those gastrointestinal issues, the stomach issues over a long period of time do persist, even though they made them better since the Boulevard case. It's still not perfect. Opioids are really reserved for they should be reserved for at least the worst of the worst. Obviously, the United States.
Like should be is the key word there.
Should be, yeah, exactly. And I don't think I need to tell your audience what the repercussions of that are. So
please do.
So what we're faced with is really a lack of options. And from what we've seen, psychedelic, there's a major mental component to pain. And from what we've seen, both 5-HT2A and the 5-HT2C receptor sites should have application in treating these chronic pain disorders. One which I think is of particular interest would be cluster headache. Another one would be chemotherapy-induced neuropathy. So there are a number of applications, chronic pain applications, which we could potentially use a drug for. And right now we are working with the National Institutes of Health, which is a U.S. government organization, to test Bright Minds compounds in various pain indications to see whether that application would hold true. And we're continuing on that work, and what we've seen so far is encouraging.
So let's say for a second that I have a chronic pain issue. Would this be something that I would be taking daily to prevent some sort of flare-up, or would it be something that I'm taking like when I feel like a stabbing pain or whatever? And also, would this be a psychedelic effect that I would then be feeling, or it's non-psychedelic?
It's a complex question. There's really a lot of ways we can tackle that, and it's going to depend on the indication. So why don't we just pick one with cluster headache? So cluster headache, it could both be used as an abortive and a preventative. In practice, how you would use it would be as a preventative, meaning that you would take, call it a medium-ish dose of the psychedelic drug, and you would have protection from cluster headaches for a period of time, probably around a month. Obviously, more work needs to be done on exactly the dosing regimen, but that would be one application there. And there's potential to, with biased agonism, potentially have that therapeutic effect without a full psychedelic effect, perhaps a lessened psychedelic effect or no psychedelic effect.
Okay. Yeah, I guess that would be probably ideal. I mean, as much as some people would like to have a full-on psychedelic event, it's not the most practical at times.
Yeah, and I mean, there's those psychedelic drugs. If you love the feeling, there's nothing stopping you from ingesting those. But for some of these people who may be your grandma or your aunt who never touched drugs and would never think about touching drugs, it may not be appealing to them. So to really treat patients on a broad scale, we're looking to create drugs that have that therapeutic action without something that could be considered a liability from somebody taking it or prescribing it.
And something great about this that I just really want to nail the hammer on the head. I think I said that wrong, but whatever. The that we kind of touched on was if we can have a better system of treating severe pain, then we can, at least to a certain extent, somewhat circumvent the opioid addiction crisis, because a lot of the people that are ending up getting addicted to these drugs, they're originally prescribed an opioid from a doctor. It's not like they went out on the streets the first time and they're like, I need opioids. It's they feel these effects because they have chronic pain issues, because they have fibromyalgia, because they are going through chemo, because whatever, they get prescribed opioids and then they just can't get off of them. So if we can circumvent that first instance ever happening in the first place, then hopefully we could put a solid dent into the opioid crisis.
Yeah, that's exactly right. And as you very eloquently said there, it's imperative, I think, to create these new alternatives to the aspirins and fentanyl. You need something in between. And as you said, it's somewhere between 8 and 9 out of 10 people in the United States that end up with a fentanyl or severe opioid addiction start with prescription pain medication, which, I mean, if doctors had a safer alternative, they wouldn't be prescribing those killer drugs.
Mm-hmm. Well, I mean, there's also the issue of some doctors being basically paid into over-prescribing opioids, but that's a different story. So I guess moving a little bit, one of the most interesting aspects of your indications, the one that kind of, to me at least, feels like the most out of left field or whatever, I don't really understand how, or don't really know how to understand it, is treating epilepsy or other types of seizures. So how does that really work? Because that seems to me like completely mind-blowing, the idea that could use drugs like this to help treat seizures.
So that drug would be working on the 5-HT2C mechanism, not 5-HT2A, meaning that it would not be a psychedelic drug. It would be a regular daily prescribed pill. And we have we've seen very encouraging evidence in preclinical models. We have seen patient data from drugs that are less selective in their approach, but give us confidence that the biology, that 5-HT2C mediates that epileptic action. And in our preclinical animal models, we've proven that, at least at the animal level. So we're very excited to go into those indications. Again, we think we have a de-risk mechanism and a best-in-class drug.
How does that even work? Do you have to find, this is kind of a little bit of a tangent, but I'm interested. Do you have to find animals that have epilepsy, or are you able to induce epilepsy in animals? Like how it would seem to me it would be hard to find a proper population.
Yeah, so there's a couple different ways, and you touched on, I guess, the two big ones. So hypothermia-induced seizures would be one route. In other models, they're shocked, and then there's spontaneous seizures, which would be selecting for a population that's susceptible to seizures or seizure prone.
Makes sense.
So there's a few different ways to do it, and each have their benefits and drawbacks. So we've tested it in a number of those and are quite confident in our findings to date.
Awesome. So I think that's a good segue to get into the last topic that I want to talk about, which is clinical trials. So I know that you guys are in the late preclinical stages on quite a few different compounds and indications that you're looking to treat. So I guess my first kind of question on this topic would be, what indications do you think will be the first ones to make it to a phase 1 clinical trial? And when do we expect that to happen? Is it still like another year or two, or what's the kind of timeline?
We'll be in phase 1 clinical trials early next year. We'll be in phase 2 clinical trials late next year.
Exciting. Congratulations.
Thank you. Thank you. So we'll be in phase 1 clinical trials with our drug to treat epilepsy, with our drug also to treat Alzheimer's disease psychosis, and we're deciding on potentially doing a third indication there as well.
Are
So
Alzheimer's and psychosis, those would be the C transmitter, or they're psychedelic for the A?
they would be the C
They
transmitter.
both be the C transmitter? Okay.
Yeah, yeah.
Okay.
We've run our compounds in schizophrenia models, which is psychosis, and majority of the population who have Alzheimer's and Parkinson's develop psychosis, and this psychosis is really where they appear to go off the rails in terms of auditory hallucinations. Sometimes they'll think that people are living in their basement or the government's out to get them. Mm-hmm. And this activity is really tough in the family and often results in unruly activity and disrupting the whole household. So much so that unfortunately a lot of these people are meant to be put in a home, which is a really tragic way to go at the end of life. So what we believe our drugs can do is treat those symptoms. Now, on top of that, we're also looking at some of our other drugs, and this is much earlier, but looking at the potential of some of our drugs for Alzheimer's and treating that neuroinflammation. Company you may know called Lucius is working on this same concept, and we believe there is tremendous potential for this chemical class of drugs to treat Alzheimer's and inflammation.
I mean, it would be amazing if could even have an improvement, like let alone a full-on cure. As anybody that's lived with somebody with some of these issues can attest to, it can be really difficult, not only for the person going through it, but for everybody around them also. So I definitely am wishing you guys the best in those endeavors, because again, any sort of improvement in that area would be just a massive quality of life improvement for everybody involved. So do these 5-HT2C receptors that you would be giving, would it essentially be helping to re-regulate the chemical balance that is in these people's brains? Like, I'm assuming they're getting stimuli in these receptors that they shouldn't be getting, and that's causing part of the issue. So is this kind of helping regulate it and flatten out some of the chemicals that are active in the brain?
Yeah, in its basic form, there's a serotonin imbalance, and that's true with not all cases of mental health disorders, but a lot of them. That's why SSRIs work for a subset of the population who have depression. Really, with the more, I'd say, deeply affected part of the depression population, that's where psychedelics really kick in. The people with suicidal. Yeah. So, it's really a dysregulation of the function.
Perfect. And then last question on clinical trials. The other two areas that you have on your website where you're in preclinical are PTSD and depression. Do you expect a phase 1 to happen in 2022 for those, or that's still more 2023? Yes.
Nope, we're targeting 2022. So
okay, so it looks like 2022 is going to be quite a pivotal year for you guys. You've got, what was it, at least 4 different phase 1 trials, and then hopefully two phase 2 trials by the end of the year.
Yeah, we've got. I don't want to. We probably won't run 4 trials with our 5-HT2C drug, but we will run multiple trials. So we have those coming up, and then very excited about our psychedelic drug program. I think.
Well, it sounds exciting. Like just the base philosophy of kind of moving past the classical psychedelics and already starting the work on the next generation drugs, and then also, of course, the ones that aren't psychedelic as well, really could pay off in a few years once all of these companies that have been putting their resources into classical psychedelics for 4 or 5 years, all of a sudden they're going to be competing with newer drugs that don't have some of the downsides that classical psychedelics do, assuming, of course, these clinical trials go well.
Yeah, that's the idea. And I mean, we're really major fans of all these first generation companies. There's a lot of work that needs to be done in terms of in terms of not only getting the general population more comfortable with these drugs and knowing that these treatment alternatives do exist, but also getting the more broad medical establishment and the infrastructure set up to roll out these second generation drugs, which I think we're all moving in the right direction and on the same team. So definitely very happy to see that those drugs will be on the market in the relatively near future, treating patients that
maybe even like as soon as, what is it, late next year, maybe MDMA, or is it still 2023 they're expecting?
You'd have to ask Rick Dov on that, but
soon enough.
I'm hoping I'm hoping very quickly. So
one last question for you, actually. I just thought of it now. So long term, what is the vision for Bright Minds? Do you vision yourselves mostly as a pharmaceutical company that's just producing the drugs and then selling it to whoever, the pharmacy or the clinics or who be it? Or do you ever see yourself getting into that side of the business yourself? Or are you just strictly a pharmaceuticals company long term?
So Bright Minds is at its core really based on innovation and the basic science, creating these new molecules, understanding how they work in the body. And then from that point, we are also good at early phase de-risking. So the preclinical animal testing, and then after that, the early phase clinical. The phase 3 clinical trials, the commercialization strategy, we feel is probably best left to an established pharmaceutical company that has those sales forces.
So you would license out the molecules, essentially?
Pardon me?
You would license out the molecules? Okay.
Yeah, we would certainly consider that. I think another potential scenario for us would be a large pharmaceutical company that's looking to get interested in the space and doesn't have the in-house scientific capabilities. And by the way, none of these guys do, because this was completely outside of their interest level. If you had a 5-HT2A drug, it was a liability and you completely dropped it. You wouldn't look at that compound anymore. Mm-hmm. So it could be a corporate takeout and somebody trying to acquire not only our pipeline but our scientific expertise and excellence in those fields. So I think we're going to stick true to our knitting and really keep turning out these new innovative compounds and looking for new treatment areas to utilize these compounds in, such as Alzheimer's would be an example. We've actually created some 5-HT2B inverse agonists, which basically they block the receptor. And we've run some preclinical trials in cancer treatment. So running it through various potential cancers. And we've shown
to treat the pain of the cancer or to treat the cancer itself?
No, to treat the malignancy, to treat the actual cancer.
Okay.
So we've seen very encouraging results. It's still early days in that, but there's things like that we're doing. I think that sort of probably gives your audience an idea of the capabilities of our team. And
well, perfect. I've enjoyed this conversation immensely, Ian. Is there anything else that you want to get out to any viewers or investors or potential investors before we end the interview?
If you guys have any further questions, always feel free to reach out to me or anyone else in the company if you'd like to.
You want to throw out an email or Twitter or something that people can reach you at?
Yeah, sure. Ian at Bright Minds Bio.
Ian at Bright Minds Bio. Perfect.com.
That's it.
Perfect. Well, it was a pleasure. Thank you so much for being on the show today, Ian.
Thank you so much. Appreciate it.
Bye-bye.